Imagine your body as a heavily fortified ancient castle. Every single day, literally every hour, it's under constant, invisible siege by millions of microscopic invaders. Viruses, bacteria, fungi, they're all trying to breach the walls and plunder the resources inside. Okay, let's dive into this, because how we survive this never-ending microscopic war is nothing short of miraculous. In this explainer, we're going to look at the definitive schematic of our biological defenses. We survive in a world absolutely teeming with microbes solely thanks to a brilliant, highly structured cellular fortress. Over the next few minutes, we're going to break down exactly how this machinery recognizes foreign invaders, mounts a really precise counterattack, and then, astonishingly, remembers the enemy for decades to come. Here is our blueprint for today. 1. 2 arms of animal immunity. 2. Innate defenses and local alarms. 3. Adaptive immunity and specific recognition. 4. The coordinated cellular counterattack. 5. Memory and building immunity. And finally, 6. System failures and evasion. Section 1. 2 arms of animal immunity, the fast grunts versus the slow elites. Now, what's really interesting here is the clear distinction between the 2 cooperative systems protecting you right now. On one side, we've got innate immunity. This is your rapid, general response. It's active immediately and recognizes broad traits shared by groups of pathogens, but it has no memory. On the flip side, we have adaptive immunity. Found only in vertebrates, this is a much slower response that develops after infection, but it's highly targeted to specific pathogens using a vast array of unique receptors. And crucially, it remembers. Section 2. Innate defenses and local alarms, the walls and the first responders. Before any cellular battle even begins, invaders have to get past the moat and the castle walls. Your skin provides incredible physical armor, and its low pH of 3 to 5 prevents a whole lot of bacterial growth. If microbes try to enter through the respiratory or digestive tracts, well, mucous membranes produce a viscous fluid to physically trap them, and if they ride in on your food, your stomach acts as a deadly chemical moat, using a highly acidic environment to destroy most microbes instantly. But what happens when an invader actually slips past those barriers? That's when patrolling phagocytic cells step in, like neutrophils and macrophages, which literally translates to big eaters, by the way. They act like biological Pac-Man in a really methodical 3-step phagocytosis cycle. First, the cell detects the pathogen and extends these little arms called pseudopodia to engulf it. Second, the pathogen gets trapped in a vacuole, which then fuses with a lysosome packed full of toxic enzymes. And third, the pathogen is completely destroyed and the debris is just expelled. Now, think about getting a splinter. It instantly hurts, turns red, and swells up. That is a localized inflammatory response. When the skin is breached, it's a frantic race against time. First up, the alarm. Mast cells in the tissue release a chemical called histamine. Second, the swarm. This histamine causes nearby capillaries to rapidly dilate and become permeable, flooding the area with fluid and a swarm of hungry neutrophils. And finally, the cleanup. Those neutrophils digest the pathogens and cell debris, paving the way for the tissue to heal. Section 3. Adaptive immunity and specific recognition, espionage and elite intelligence. To understand the elite forces, we really have to define their primary target. An epitope is the small, accessible portion of an antigen, which is basically any foreign substance that binds directly to a specific antigen receptor. This is the absolutely crucial molecular puzzle piece that our elite lymphocytes are trained to hunt down and identify. And this all relies on a strict lock-and-key model. Your adaptive immunity relies on B cells, which mature in the bone marrow, and T cells, which mature in the thymus. These cells possess millions of unique surface receptors. You see, they aren't just looking for general bacteria, they're looking for a perfect, exact physical fit with the epitope of a very specific invader. And crucially, through a process called clonal selection, once a single lymphocyte finds its perfect match, it rapidly clones itself to build a massive targeted army. However, B cells and T cells have very different methods of gathering intelligence. B cells are capable of binding directly to intact, free-floating antigens right in the extracellular fluid. T cells, on the other hand, are effectively blind to free-floating pathogens. They can only bind to antigen fragments if those fragments are actively presented on the surface of a host cell by something called an MHC molecule. Section 4. The coordinated cellular counterattack, the two-front war. So the really crucial point here is that this two-front war doesn't just happen in isolation. It's commanded by a single linchpin, the helper T cell. When a helper T cell recognizes a presented antigen, it acts exactly like a commanding general. It sends out chemical signals that simultaneously trigger the activation of B cells for humoral immunity and cytotoxic T cells for cell-mediated immunity. Let's look at those two fronts side by side. The humoral response, driven by B cells and antibodies, secures the extracellular fluids, the blood and the lymph. It neutralizes toxins and pathogens before they can even enter host cells. Meanwhile, the cell-mediated response, driven by cytotoxic T cells, operates intracellularly. It specifically hunts down your own body cells that have already been compromised and infected by the pathogen. Let's move to the humoral response and see how this builds. A really common misconception is that antibodies kill pathogens directly. They actually don't. They're basically molecular tags that mark the enemy for disposal. They can physically blockade a virus from binding to a host cell, a process called neutralization. They can coat a bacterium in a process called opsonization, creating a beacon that massively accelerates phagocytosis. Or they can trigger the complement system, which literally punches structural pores into the foreign cell, causing it to just burst. Over on the cell-mediated front, it's all about targeted destruction. When a cytotoxic T cell binds to an infected host cell displaying a viral antigen, it unleashes a brutal chemical attack. It releases perforins, which form pores in the infected cell's membrane, and granzymes, which enter those pores to trigger apoptosis, which is programmed cell death. The infected cell fragments, and that successfully halts viral replication right in its tracks. Section 5. Memory and Building Immunity. The Veteran Reserve Force. Now, this activation curve is perhaps the most important concept to take away from this whole explainer. Look at the stark mathematical difference here. Upon initial exposure to a pathogen, the primary immune response is slow, taking around 10 to 14 days to really peak. You get sick during this time. But because the adaptive system creates long-lived memory cells, a secondary exposure to that exact same pathogen months or even years later triggers an immediate, overwhelming antibody strike. You often fight off the infection before you even feel a single symptom. It's incredible. We can actually leverage this memory through vaccination. Active immunity, whether acquired naturally through getting sick or artificially via a vaccine, forces your body to generate its own immune response and memory cells, providing protection that can last for decades. Passive immunity, on the other hand, is when you receive pre-made antibodies, like a baby receiving them from its mother, or, say, through an antivenom injection. It provides immediate defense for sure, but no memory is created, meaning the protection degrades over a few weeks or months. Section 6. System Failures and Evasion. When the fortress falls. It begs the question, right? For all this precise lock-and-key recognition and coordinated warfare, what happens when this perfect defense system gets confused? Or worse, what happens when it gets actively outsmarted by an evolving enemy? But we actually see both scenarios play out. First, consider hypersensitivity or allergies. This is the system getting confused. Harmless allergens like pollen trigger unnecessary histamine releases from mast cells, causing the vascular dilation and downright misery we know as hay fever. But an even greater threat is immune evasion. Pathogens like influenza outsmart us by constantly mutating their surface proteins, a process called antigenic variation. Your existing memory cells are looking for yesterday's keyhole, rendering them totally useless against today's mutated virus. And some viruses go even further. HIV, for instance, uses latency to hide its viral DNA directly inside the chromosomes of our own host cells, making it completely invisible to the immune network until it decides to strike. The evolutionary arms race never truly ends. Our cellular fortress is an absolute marvel of biological engineering, but the microscopic invaders laying siege to it are constantly adapting. It leaves you wondering, in the invisible war raging inside us today, who is truly winning? Keep learning, stay curious, and we'll see you on the next explainer.